this gyrus up here, singular gyrus, this structure, this ventricle, or lateral ventricle. All right, what structures make up the inside wall of this ventricle? The caudate, right, the caudate. And then what's going to be behind the caudate over here? So the caudate is medial, just as this structure is, but then it moves out laterally to avoid running into it, and this is the thalamus, right? Below the thalamus is going to be the hypothalamus, and then the, you started to say it? Mesencephalon or midbrain, the pons, medulla, right. Okay. All right, let's go over to this model. Somebody asked me the other day during the test, is a neuron a cell? What is the answer to that question? Yes, a neuron is a cell, right? That's our take-home message for the day. All right. What are these, huh?
It's the largest cell in our body.
Oh, a larger?
Largest cell in our body or
No,
no?
the Purkinje cell is one of the largest cells, yeah. Doesn't have a, then you have projection ones from the cortex going down the spinal cord, and those would be longer ones, but maybe not larger, right? Okay, what's this representing? Dendrites. And then this is the soma or cell body. It's going to taper off into the axon hillock, which is going to transition into the axon, right? If what's this stuff over here? Myelin. If this were a peripheral nerve, it would this cell that's myelinating would be a Schwann cell, and if it's in the central nervous system, a oligodendrocyte, right? What kind of channels do we have on dendrites? Chemical gated, right? And the axon hillock is special because it has a high density of voltage gated channels, right? What's the space between the myelin sheaths called? Nodes of Ranvier, and they have a special property similar to the axon hillock, that they have a high density of voltage gated channels, right? In the axon terminal, which isn't really represented here, what's going to be contained within the axon terminal? The what's going to be inside of there? Neurotransmitters inside of synaptic vesicles, right? Now those neurotransmitters are made where? In the Nissl bodies, right? And then they'll get released, and what's going to trigger the release of neurotransmitter? Calcium coming in from depolarization of the nerve impulse, right? And then that'll contact dendrites or muscle or gland tissue, right? Okay, good with that one. Let's go to this one. So, what is this upside down? Maggie, check. With the corpus callosum, what's this membrane? The septum pellucidum. Right. So this structure is going to be sitting in here. So if we rip out the septum pellucidum, we would be looking into what? What lateral ventricle, right? If we go all the way through the lateral ventricle, we'll see a structure that makes the inside wall of the lateral ventricle. The structure is called the caudate, right? We also have this